Bacillus subtilis strains bearing translational fusions of mismatch repair proteins fused to GFP are streaked out on selective media. The streaked cells are used for fresh inoculum into liquid culture. Cells are grown until OD600 is 0.3 to 0.4, whereupon mismatch-inducing treatments are applied if desired, and cells grown further until reaching OD600 of 0.5 to 0.8. Cells are removed, the membrane stain FM464 is added if desired, and cells are applied to slides with prepared agarose pads and allowed to settle. After removal of excess media by aspiration, a cover slip is applied and cells are visualized by microscopy.
Hi, I'm Dr. Andrew Klockow from the lab of Dr. Lyle Simmons in the Department of Molecular, Cellular, and Developmental Biology at the University of Michigan, Ann Arbor. Today, we will show you a procedure used to visualize translational fusions of the DNA mismatch repair proteins as well as other repair and replisomal proteins in Bacillus subtilis. We use this procedure to study the interaction between the DNA mismatch repair proteins and the replisomal proteins in Bacillus subtilis live cells. So let's get started.
To begin this procedure, prepare the B subtilis strains containing the translational fusion protein you wish to visualize. For some strains, starting 1 day prior to imaging is sufficient. Examples of these include strains that grow well when the translational fusion between GFP and the gene of interest is incorporated into the gene's endogenous locus, such as mutL-GFP. When starting 1 day prior to imaging, use a sterile stick under sterile conditions to apply the bacteria to selective LB agar medium on plates in order to grow single colonies. Incubate overnight at 30 degrees Celsius. The next day, the colonies will be used for a starter inoculum. Some B subtilis strains require 2 days of growth prior to microscopy for high-quality images. Specific strains that require 2 days growth are those that grow poorly due to the translational fusion, such as Rec-A-GFP. On the first day, streak the bacteria on selective LB agar medium and incubate overnight. On the second day, perform a serial dilution. First, pick a single colony to inoculate 100 microliters of 0.85% saline. Then perform 4 10-fold serial dilutions. Plate 100 microliters of each saline dilution, as well as the starting colony in saline, on LB agar plates containing selective antibiotics. Incubate the plates overnight, usually at 30 degrees Celsius. After the second overnight incubation, select the dilution plate which has light confluent growth of cells. These provide a healthy, exponentially growing starter inoculum, potentially yielding excellent imaging results. This is particularly useful for the highest quality imaging of membrane staining with FM464. On the morning of imaging, place 10 ml of S750 medium into a 125 ml sterile flask, thus maintaining at least a 10-fold air-to-medium ratio. Resuspend the cells in up to 2 ml of S750 medium from the LB agar plate with single colonies or the light confluent cells to obtain an inoculum. Add enough S750 medium cell inoculum to the Erlenmeyer flask containing 10 ml of S750 medium so that the culture has an initial OD600 of about 0.08 to 0.1. Next, grow each culture at the appropriate temperature for the B subtilis strain to be imaged. Shaking water baths are preferred with revolutions per minute from 150 to 200. Continue growing the cultures until they reach an OD600 of about 0.5 to 0.8. For cultures challenged with DNA damaging or mismatch-inducing agents, plan the treatment such that the end of the incubation period with the DNA-altering agent coincides with the time of reaching the target OD600. For example, 2-aminopurine, a DNA-based analog which mimics a mismatched base when incorporated into the genome, requires 1-hour incubation and leads to focus formation of mismatch repair proteins such as mutL-GFP. However, Rec-A-GFP focus formation requires a 1-hour incubation with mitomycin C or some other DNA damage reagent for induction of double-strand DNA breaks. Both 2-aminopurine and mitomycin C are added when the cultures reach OD600 of about 0.3 to 0.4, and after the required 1 hour of growth with either treatment, the cultures will most likely have reached an OD600 of 0.5 to 0.8 and are ready for imaging. Start by pipetting 300 microliters of cultured cells into a 1.5 milliliter microcentrifuge tube. If imaging of cellular membranes is desired, add a 1 to 1000 dilution of 1 mg per milliliter FM464 membrane stain to each sample. A titration of FM464 may be required to achieve the desired fluorescence signal. A typical titration range is 1 to 100, 1 to 1000, and 1 to 10,000. Allow the samples to sit for up to 10 minutes or while the slide is being prepared. If concentration of the cells is required, filter the cells using a 0.2 micrometer filter with a vacuum apparatus. Then gently wash the cells off the filter with appropriate buffer or medium. While the cell samples are incubating with FM464, prepare 1% agarose with 1x spizizens by adding 5 ml of 10x spizizen stock to 45 ml of distilled water, along with 0.5 grams of agarose, and microwaving to melt. Let the temperature of the agarose equilibrate in a water bath to approximately 65 degrees Celsius, and draw 20 microliters of spizizens with 1% melted agarose into a pipette tip. Agarose that is too hot is difficult to pipette, and cold agarose will solidify and not enter the pipette tip. Next, use a multi-test 15-well slide and pipette a single drop of approximately 1 to 2 microliters of the agarose solution into each slide well to form shallow agarose pads. The agarose will solidify immediately. The slides are prepared immediately prior to application of the cells to avoid dehydration, which would make the slides unusable. The agarose pads should be centered and fill each well, but have minimum height to avoid disrupting the cover slip. If droplets are too high or bubble-like, or if the sample spreads beyond the well boundary, simply swipe the well clean with a Kim wipe and apply a new agarose pad. Now that the slide is ready, apply the entire 300 microliter sample, which was previously set aside in the microcentrifuge tube, to the slide, with the sample distributed on the top of each agarose pad. This amount of cells should be sufficient to cover each well on the slide. Allow the slide loaded with cells to sit at room temperature for approximately 10 minutes. This step will allow for the cells to settle on the agarose pad and become immobile, and therefore ready for imaging. At the end of the 10-minute incubation, aspirate away the excess medium from each well without removing the pads themselves. Apply the cover slip to the slide, pressing firmly and evenly, but gently, across the area of the slide. Check to make sure that the cover slip is against the slide and that the cover slip is not raised above the slide. Proceed to imaging. Many different fluorescence microscopes will work well. It is critical to have a 100x oil immersion lens. The Simmons lab uses an Olympus BX61 microscope equipped with 1.45 numerical aperture 100x oil immersion objective lens suitable for total internal reflection fluorescence microscopy, or TIRF-M. The microscope is also equipped with a CCD cooled camera and a Lumen 200 arc metal light source. Start by bringing the cells into focus using white light. Use the appropriate filter for each fluorophore. For detection of GFP, use filter excitation 460 to 500 and emission 510 to 560. For detection of FM464, use filter excitation 510 to 560 and emission 572 to 648. Capture the image using the appropriate filters for each fluorophore with the Slidebook 4.2 software. Here are some representative results of imaging the DNA mismatch repair protein mut-L, which has been translationally fused to GFP. The untreated control culture of mut-L GFP shows minimal foci, with most cells containing only cytoplasmic levels of the mut-L GFP fusion protein. However, the majority of cells grown in the presence of 2-aminopurine, which mimics a mismatched base when incorporated into the genome, have GFP foci, indicating the mut-L GFP protein is involved in recognition and repair of the mismatched DNA bases. Similar results are obtained with the Rec-A GFP strain grown in the presence of the DNA damage-inducing drug mitomycin C. Here, the majority of cells contain a focus of Rec-A GFP upon treatment of mitomycin C, indicating the Rec-A GFP has responded to the DNA damage, initiating the repair process. Without mitomycin C treatment, few foci are seen.
We've just shown you how to visualize repair proteins in live Bacillus subtilis cells. When doing this procedure, it's important to remember to take your time and bring the cells into proper focus. And doing so may actually take some practice. So that's it. Thanks for watching, and good luck with your experiments.